Our first speaker today discovered a key element to understand growth, disease, and aging. Michael Hall is professor of biochemistry at the Biozentrum of the University of Basel. He studied at Harvard and Berkeley before moving to Switzerland, where he made one of the most transformative discoveries in cell biology, the mTOR signaling pathway, which controls growth, metabolism, and aging. His work has had profound implications for understanding cancer, metabolic disorders, and longevity research, earning him numerous international awards, including the Breakthrough Prize in Life Sciences. Please welcome Professor Hall, who will speak on mTOR signaling in growth, metabolism, and disease.
So we work on cell growth, and particularly work on cell growth control by mTOR signaling. And what I'd like to do today is tell you one story from the lab. It's a story which is not yet complete. We're about halfway through, so don't expect any. It's more of a progress report than anything, and it deals with the subject of mTOR complex 2 and sensory neurons in adipose tissue. Those are very appropriate that I'm speaking directly after lunch. Okay, so before I tell you, go into this story, I'd like to give you a brief introduction to mTOR signaling. And this here is the very first ever model of mTOR signaling published. It's one we published shortly after our discovery of mTOR in the early 1990s. I have to say, I'm actually embarrassed we published this model. There's almost nothing of value there. It looks more like a Gary Larson cartoon than a real scientific. Who here remembers Gary Larson, by the way? Oh, okay, that's quite not bad. Yeah, okay. Now, we know significantly more about the mTOR signaling network. Even this picture is out of date. We no longer draw models of mTOR signaling in 2 dimensions. And what is driven this increase in complexity is this exponential increase in publications in this area. And this has been driven by the fact that mTOR turned out to be both fundamentally and clinically important, much more so than we suspected when we first stumbled across it back in 1991. So mTOR stands for target of rapamycin. This is rapamycin. This is a natural molecule isolated from a soil bacterium, which was first identified in Rapa Nui, hence the name rapamycin. And it now has applications in 3 major therapeutic areas. It's first became very famous as an immunosuppressive drug for transplantation patients. Now it's even more famous as a anti-cancer drug. It's also used on occasion for treatment of cardiovascular disease, angioplasty, restenosis after angioplasty in particular. So what is mTOR? mTOR is a kinase. It's a very highly conserved kinase, but more importantly, it's a so-called central controller of cell growth, and it controls cell growth by controlling a large number of cellular processes which collectively determine mass accumulation and thereby cell size. And these cellular processes can be divided into 2 groups: the anabolic processes which mTOR activates, and the catabolic processes which mTOR inhibits. So what mTOR does, or mTOR, it balances these counteracting forces of synthesis and degradation such that the cell accumulates the appropriate amount of mass relative to what nutrients are available. Now, how does mTOR actually control all these cellular processes? Well, here is a rather simplified overview of what we call the mTOR, in this case mTOR, it's mammalian mTOR, mTOR signaling network, and we call it a network because it's more than a single pathway. It's actually 2 signaling pathways. The defining feature of the network are these 2 complexes here, mTOR complex 1 and mTOR complex 2, each of which phosphorylates its own substrates to regulate its own cellular processes. So mTOR is in fact 2 structurally and functionally distinct kinases. Like mTOR itself, which is highly conserved all the way from yeast to mammals, the 2 complexes are also conserved all the way from yeast to mammals, as is the architecture of the entire network. So the picture that is emerged is that this is a primordial or ancestral signaling network, which has been conserved throughout eukaryotic evolution to control this very fundamental process of cell growth. And the only exception to that statement I just made is this part of the network up here, which is the growth factor or insulin signaling pathway, which evolved later. This evolved multicellularity and was then grafted onto the more primordial mTOR signaling network. And the reason for this is that growth control in metazoans and multicellular organisms is more complex than cell growth control in unicellular organisms such as yeast, because in multicellular organisms, it's key to control the growth of every cell in the body relative to every other cell in the body, so that our organs end up being properly proportioned. And that then is what the growth factor signaling pathway provides. This provides a growth signal over a whole body plan, combined now with local nutrients, amino acids in particular, and now we also know energy. So these are the three conditions which upregulate mTOR signaling: is energy, nutrients, and growth factors, at least in metazoans. So another fascinating aspect of the mTOR signaling network is the unusually large number of diseases to which it is functionally linked. And what all these diseases have in common is that they are characterized by inappropriate or ectopic cell growth. And these can be benign forms of cell growth, or malignant forms of cell growth, or benign forms of cell growth, but in all cases, cell growth. More recently, mTOR has also been implicated in so-called metabolic disorders, obesity and diabetes, and this is what I'll be talking about later, role of mTOR in diabetes. So this is the talk I would like to give you now. Deals with mTOR complex 2, one of those two conserved complexes in the mTOR network, in this case, specifically in adipose tissue. So this story actually starts a little over 15 years ago when we made an adipose-specific knockout of the Richter gene, which is a essential and specific subunit of mTOR complex 2. So we knocked out mTOR complex 2 specifically in adipose tissue. And what we found was that these mice became hyperinsulinemic and insulin resistant. So they developed systemic metabolic defects. And this is part of the non-cell autonomous function of mTOR, at least in this particular tissue. And this led us to propose a model where mTOR controls systemic metabolism by signaling to peripheral organs, in this case, liver and pancreas, to control IGF insulin production. And the logic behind this mechanism is it provides a mechanism of insulin homeostasis. And this is due to the fact that mTOR complex itself is activated by insulin and IGF. So when these become too high in the bloodstream, they can activate mTOR complex 2 and then shuts down their production by signaling to these other tissues in a negative manner. Now, we spent many years, we were quite excited about this model because it provided a new way of looking at insulin homeostasis. And we spent a great deal of time trying to understand what this factor was, this TORC2 dependent secreted factor, which would act on other tissues to control systemic metabolism. We actually spent about 10 years looking for this factor, always failing. It was the kind of the rite of passage for every new postdoc who came to the lab to make an attempt to find this factor, and we always fail. And the reasons for that should become apparent soon. But recently, a student by the name of Irina Fry decided to go back and repeat this experiment, but because there was something else wrong with this experiment, and that was the Cree driver we used is expressed at birth, and fat does not become fully developed until 6 weeks of age. So we were afraid that what this systemic defect could be a developmental defect. So she wanted to delete mTOR complex 2 in the adult. So she did that by using now another Cree driver, the adiponectin Cree driver, which is tamoxifen inducible, to knock out again the Richter gene, which is this subunit of mTOR complex 2. So she treated adult mice in this case with tamoxifen to induce a knockout, and then harvested mice at different time points at 3 days, 2 weeks, and 4 weeks. And what she found was that much like we'd found 15 years earlier, that these mice also became hyperinsulinemic, high levels of insulin. You have the control in white here, and then this is 3 days, 2 weeks, and 4 weeks. You can see at every time point, these mice are hyperinsulinemic, and we look at insulin resistance here as measured by an insulin tolerance test. And as you can see, in this case, at the 3-day time point, the mice are still sensitive to insulin, but now they develop insulin resistance at later time points. So the good news was that we'd reproduced what we'd found earlier, telling us that these systemic metabolic defects are not developmental defects. So Irina wanted to understand what was wrong in this adipose tissue, lacking mTOR complex 2. So she isolated adipose tissue, control and knockout, did mass spec, analyzed the data, and what she found, the proteins which were most commonly changed were membrane proteins, and in particular, neuronal synapse proteins, which of course surprises somewhat because we thought we were working with adipose tissue. But as you probably all know, adipose tissue is highly innervated by the sympathetic network, which is an efferent system which signals from the brain to the adipose tissue. So Irina developed a method to look at the sympathetic network, nervous network in adipose tissue, looking at 3D whole mount images. And here's what she found when she imaged the sympathetic nervous system in this fat depot. She found a very intricate network where the nerves came in from the, at this end, followed blood vessels, and then branched out to into the parenchyma of the adipose tissue. She then looked in the knockout mice to see how this might be affected, and much to our surprise, she found absolutely no effect whatsoever. The sympathetic nervous system was perfectly normal in the TORC2 knockout compared to the control, telling us that whatever neuronal proteins were changing, it couldn't be in the sympathetic nervous system. And this then really confused us until we delved into the literature and found this work by a researcher by the name of Tim Bartness, who is a neurobiologist who worked at Georgia State University. He passed away about 10 years ago, and he had done experiments using the tools of his day, which led him to argue that there is a sensory network in adipose tissue. Unfortunately, his work never gained traction. Nobody ever pursued this work, but at least there was this indication that there was perhaps sensory networks in adipose tissue. Of course, the difference between sensory and sympathetic is in the case of sensory, they signal from the tissue back to the brain. I myself do not know there might be sensory neurons in fat tissue. And most of the people I talked to had no idea about this either. It seems sort of somewhat counterintuitive that fat should have sensory neurons. What, for example, could they possibly be sensing? We also think of adipose tissue as a transient tissue. So why would it have a developed sensory network? So be that as it may, Irina then looked at a stain for, use a marker which will stain sensory neurons, and she found now a nicely developed sensory network in this adipose tissue as well. Not as fully developed as a sympathetic network, but certainly a nicely developed network. So this was the first ever visualization of a sensory network in adipose tissue. Arden Partaputian also recently showed there was a network and published about a week after we did. We had no idea he was working on this, and I suppose he had no idea we were working on it either. So it was one of these nice convergences of two different labs coming to the same conclusion. So if we look at this, this new kind of network, neural network in adipose tissue, and look at higher magnification, you see the ends of the neurons seem to contact individual adipocytes, suggesting that they actually might be forming synapses with these adipocytes. So this is a functional sensory network. And Christy Townsend, at Ohio State, has looked at this at even higher magnification, higher resolution, and has found that these neurons actually form a very unique kind of structure. They kind of lasso individual adipocytes. Is like a boa constrictor wraps around these adipocytes to form this what they call neuroadipose nexus. I think there are a lot of analogies between this and specialized structures we see in muscles, for example, the neuromuscular junction. So this is, I think, a lot of very new and interesting biology going on here. So the question, of course, is, does TOR complex 2 affect this sensory network? So this is the experiment done by Irina. Again, the control here, we see this network forming. But if she now looks in the 4-week-old, the mice in which TORC2 had been knocked out for 4 weeks, she finds this bundle of neurons coming in from the dorsal root ganglion, comes in along the blood vessel, but then this bundle of neurons doesn't branch out, doesn't arborize to innervate the parenchyma. So for somehow the nerve endings seem to have retracted or shrunk back into this the nerve bundle. These nerves don't go apoptotic because the ends of these nerves, which are in dorsal root ganglion, are still intact. So somehow this is a retraction of the nerve endings in this adipose tissue. Just to illustrate that the sensory and the sympathetic are indeed 2 separate networks, Irina did a double staining. Here you can see the neurons coming, both types of neurons coming along the blood vessel together, and then they branch out to form their own networks. And in the knockout, again, we see the sympathetic with little to no sensory networks. So these are really 2 very separate networks in the adipose tissue. So Irina then wanted to do the experiment to understand how is TORC2 responsible for this network? Is it responsible for establishing it or maintaining it? So she looked at this network in the mice at different time points after deletion of TOR complex 2, and she saw that at the 3 days after knockout, the network seemed to be perfectly normal, but then by 1 week, which I don't show here, there was a sort of an intermediate effect, and then by 2 weeks and 4 weeks, we lose this arborization. So TOR complex 2 seems to be essential for maintaining, at least maintaining this network. It somehow creates a signal which prevents these neurons from retracting, and this is, of course, all just quantitated down here. So what is the purpose of this? What's the physiological role of this sensory network in adipose tissue? And to get some handle on this, what Irina then did was to look at the kinetics of disappearance of the network with the kinetics of appearance of these peripheral metabolic phenotypes. And what she found was a perfect correlation between disappearance of the network and appearance of the systemic insulin insensitivity. So, which suggests that perhaps TOR complex 2 is maintaining these sensory networks, and that then is how TORC2 in adipose tissue is signaling to other tissues in the periphery to control systemic metabolism. So let me just clarify a little bit what I just said there. This is traditionally how we think adipose tissue acts on other tissues in the body to mediate whole body metabolic health. In this case, it's always by secreted adipokines, very well known as leptin, for example, fatty acids, and adiponectin. They have receptors in these different tissues to control their metabolic activity and systemic energy homeostasis. And this is the sort of the current model on how adipose tissue acts on or acts systemically. But our findings, for example, like from the Patapuchin and Ye labs at Scripps, suggest that the adipose tissue might also be signaling to these other tissues through the central nervous system. So the sensory nervous system signals back up to the brain, as shown by Bartness already years ago. He proposed that this signaling back is to negatively control activation of the sympathetic nervous system in the same tissue. So this is the way the body turns off the sympathetic activity in fat tissue, is through the signal going back. But there's more recent work showing that the sensory nervous system might be involved in signaling to these other tissues, expands this model. It's not just through a circulating soluble factor, which is secreted by the fat, but it's also now going through the central nervous system, not only to fat itself, back to fat itself, but now also to these other peripheral organs. So this is, I think, is a quite exciting finding, because it's a completely new way of looking how systemic metabolism is controlled and how diabetes might develop. It might be a problem of the central nervous system. So how are we going to pursue this? What do we want to do next? Well, what we're trying to do is, of course, trying to understand how TOR complex 2 controls or maintains these sensory neurons in the adipose tissue. And to address that issue, what we've tried to do is recreate it in vitro. So what we're doing now is co-culturing neurons and adipocytes, either in 2D cultures or in 3D cultures. In the case of 3D cultures, it's with dorsal root ganglion organoids and so-called adipoids, adipose organoids, which are just being developed now, and then co-culturing them in microfluidic devices where we can separate them in different compartments. So here is what we see when we do 2D culture. So this, again, would be this microfluidic device. Here are the neurons. This is 2D. Here are the adipocytes, and they're separated by this barrier where they're microgrooves. And so we can assay the ability of these neurons to grow, send out neurites which cross through these microgrooves towards the adipocytes. We try to recreate what we think is going on in vivo. So if we do this experiment but don't add adipocytes here, just add medium, we have some neurites extending across. But then if we add adipocytes, which are shown in red here, there seems to be a stimulation of the growth of neurites towards the adipocytes. And they actually try to search out. They grow towards the adipocytes. So there's, and this is in 2D, and with the effect we see in 3D is even more dramatic. Again, if we have medium on this side, and here we have a dorsal root ganglion organoid, we see some neurites getting across the barrier here. But now if we add this 3D adipoid or adipose organoid, almost all the neurites seem to be searching out this adipoid and presumably making these contacts I showed you before. So we're excited about this, having set up this in vitro system, because what this does is it gives us an assay to possibly purify this secreted factor, this neurotrophic factor secreted from the adipocytes, which will attract growth of neurites. And it also is a way of characterizing whatever that neurite, that factor might be. So here's just my final slide, one example of the experiment we've done using the system. So in this case, what we've done is we've cultured human adipose tissue and taken the medium, and this is of course what we call then conditioned medium. In this case, we can add the conditioned medium on one side of the microfluidic device and then our sensory neurons on the other. So here's what we see if we have just the medium, not conditioned. Some neurites get across, but not very much growth going on. If we add the conditioned medium, we see these neurites growing out, there's stimulation of growth. And then if we take this conditioned medium and treat it with a protease, which we can then inactivate, we inactivate this neurite outgrowth stimulating factor. And this is all quantified down here. So we think this diffusible neurotrophic factor is a protein. And of course, this gives us more information on what to go after. And this is exactly what we're doing at the moment. We're doing this by setting up a CRISPR screen in these microfluidic devices to see what we can knock out on the adipocyte side, which will prevent the knockout, as well as trying some more biochemical approaches. But I hope we have this neurotrophic factor identified pretty soon. So this is what I want to conclude from this the talk. So the first thing is adipose tissue is innervated by sensory neurons. I can't claim discovery of this. It was Tim Barton's discovery. I'm sorry he's not here as interest is ramping up now, finally, many years later. A number of labs are starting to jump on this. We think TOR2 and a diffusible factor are essential for this sensory innervation on the adipocyte side, of course. We also think that adipose tissue may mediate whole body energy homeostasis via this adipose CNS axis, in addition, or maybe even instead of via these secreted factors, which I talked about, of course. I think the secreted factors are playing a role. We just have to now understand which is more important, the secreted factors or this potential signaling through the central nervous system. And of course, we'd like to also translate our findings. And another thing we're very excited about is we believe that what we've found could be the molecular basis of diabetes-induced neuropathy. So diabetics often lose sensation in their fingertips and their toes. This is very debilitating. Nobody knows what the underlying mechanism is. And of course, TOR complex 2 is part of the insulin signaling pathway. So if in diabetics, if you lose signaling through the insulin pathway, you would lose TOR2 activity, and these neurons might retract, at least in the fat pads of our fingers and toes. So, of course, if this is the case, and we then identify this neurotrophic factor, perhaps we can find a therapy for this condition. Okay, that's the end of what I have to say. The work was done in 2 different labs. This is my academic lab at the Biozentrum, and this is my lab in a corporate environment at Roche. Some of these people started the project on this side and then moved to this side, but I'm just highlighting them over here. Mitsugo Shimobayashi and Irina Fry were the ones that primarily responsible for these findings. Okay, thank you very much for your attention, and happy birthday, CBM. I think 1 or 2 questions. Is that okay?
Yeah.
Okay.
We have time for 1, 2 questions, very quick. So,
yeah.
do you want a mic or?
No.
Okay.
Thank you.
Yeah.
Is the irritation of the adipose tissue something that's stereotyped and programmed during development, or does it happen in post-embryonic life in response to nutritional signals?
That we don't know yet. There are so many things we don't know. We don't know if that happens in visceral fat versus subcutaneous fat. This is obviously a wonderful question that we have to address. Yeah. Yeah.
You talked about the efferent function, the
Yeah.
afferent function.
Yeah.
Of the
Yeah.
afferent.
Yeah.
And you noted that they release things like CGRP, et cetera. Is there an efferent component to this where the sensory neuron is releasing stuff peripherally to affect?
That is also a very, we stain those neurons with CGRP, which is a secreted factor. So that's a real possibility, yes.
I assume you've asked if NGF is a relevant factor.
That is on our very short list of factors we're focusing on, yes. Yeah.
Have you looked at the receptor for the CGRP? Is that expressed throughout the tissues? Is there something selective for the adipocytes? And do you require the synapse formation in order to trigger the expression of the secreted factor?
We haven't looked at CGRP receptor. We're using that simply as a marker, but I think everything you refer to then kicks in after we've formed the synapses. It's then it gets to the issue, what are these sensory neurons actually sensing? And we're sure still, we haven't gotten there yet. We're still working on how these synapses are created. And then we can, Pataputin in a lab in Israel has recently published that piezo is somehow involved on the neuronal side. So I think there's some mechanosensation as part of the process, but my feeling is there are probably many different kinds of things. There are many different kinds of sensory neurons, and they're sensing many different kinds of things. Mechano transduction is just one part of the story. I think it's going to get much more complicated than that. And I think we're still taking baby steps. It's very early in this area.